Okay, welcome to your first unit test review session here. This would be something that you can listen to and kind of watch through passively to help you through the review guide and get you ready for our first test, which is coming up very soon here. So let's get to it. So one of the first things that we're going to address on the test is your understanding of what PPE is, which is personal protection equipment. Right, so the biggest thing is you need to know what these things would be used for, right? So goggles, obviously we want this in any situation, or glasses, that there might be any sort of splashing or possibility that things could rupture and break and things could be a projectile, right? And get into your eyes. Because if you can't see, then that causes problems with you getting to a safe location if you're in danger. Any sort of respiration type of device would be if there are things that are being spread through the air, right? So we would usually use this for things like bacteria and viruses, if you're working with bacteria and viruses. Sorry, I'm running out of kind of space here. There's a variety of different gloves. Whoa, a variety of different gloves that you could use. Could use these gloves, and we could use these gloves. These bigger ones would be for ones where you're not using a whole lot of dexterity, but the thing that you're working with might be a little bit more rough. These gloves, the ones that we've already used in class, would be for things where we don't want to possibly get into minor cuts that we would have on our hands, or if it's just a nasty material that you're working with, right? We will be using these. We used them when we were doing the blood simulation lab, but we also will be using when we do dissections, because the stuff that we're working with in dissections, it's kind of gross. It's a formaldehyde material, so it makes your hands kind of stinky. You really don't want it to get into any little cuts that you might have on your hands. Now, in the future, when you're working in possibly the medical field, you would use these gloves if you're working in surgeries or handling things where you want to keep things sterile, right? So it's not just about things getting in you. It also might be things getting out of you, right? So if you have bacteria that are on your hands, which is inevitable, we might not want to get that onto your patient. Gowns, obviously, protecting you from the environment, but also, once again, protecting you from somebody else's environment, right? If you're working with a patient. Biohazards would be if you're working with things that are life-based, right? So this would be bacteria and fungus, fungi, that's me, fungi, or viruses, right? Or even these funky things called prions, which we might talk about in the future. But all those types of things would be things that are technically mildly alive or fully alive, that can be pathogenic. We want to prevent ourselves from those things. The booties, right? If you have boot coverings, that would be if you're in an area where we can transmit it back into an area that's not sterile, right? If you're working in a location. Regardless, the big idea here is you need to understand what PPE stands for and make a logical decision about what types of equipment you would want to use in a given situation. Forensic science, right? So this is the first unit. Forensic science, we're all about collecting information from a scene of a crime, right? So this is crime scene type of situation, where you might collect, I don't know why I spell it like that, scene. Crime scene situation where you might be collecting things like fingerprints, right, where we analyze fingerprints in class. You might be analyzing blood splatter, right? This could be time of death by using temperature change. This could be, even, we don't have this simulation, I wish we did, it would be really cool if we did some entomology stuff with insects, right? You could be looking at things like hair at the scene of the crime, shoe prints, right? But the whole big thing about what forensic science is really about is taking all this stuff because somebody wasn't there to see it happen. Nobody recorded it. So we can use this information to create a story, as morbid as it is, and that story will help us explain what happened. And you can't just whimsically say whatever you want happened. You need to have support, right? So that's what the evidence does, is it supports your final conclusion about what had happened at that scene. Now, we're just finishing up right now your autopsy report, which is incomplete at this point because we're going to keep some things in the dark for you, about how Anna has perished, right? So your goal here is to create a story or an idea of how she ended up passing away, and having some evidence from your crime scene and some of the other pieces that we've given you so far about how she ended up passing. All right, so that's forensic science. That's what you would do forensic science for. This would not be something like heart surgery, right? Heart surgery is not forensic science, or um, diagnosing a person for strep throat. That's not forensic science. Forensic science is collecting a bunch of information from a scene of a crime, a lot of times, to determine what had happened, right? And so you would be helping lawyers, you'd be helping police departments, maybe even work for the police department, to figure out what happened at that location. All right, so kind of leads us into experimental design. And you got your first brush of this with the blood splatter lab. We had a question in that situation, but this is for any type of experimental design. So a scientific question would be something like why, or what, or how, right? So these types of questions are things that you would develop when you go into your experiment. So in our blood splatter lab, we were trying to figure out how the blood got at the scene of the crime. Did it get transferred from a knife? Did it get transferred from a hand? Was it impact? Was it passive transfer? Right? Based on what we kind of looked at, we understood that it was probably passive, but then we had to figure out how those spots came to be that size, right? So you developed an experiment and a hypothesis around the exact same time. So a lot of times these two go in hand. Now, with hypothesis writing, if you feel very worried about writing a hypothesis, you can go back to your old school way of doing the if, then, because. It's not bad. It's just not what is normally used in science. This is the training wheel version of writing a hypothesis. The big way that you write a hypothesis, if in the future you publish papers or design your own experiments as is, you might not even have a hypothesis, which is even a totally different story. But the big thing here is you just have to be very definitive. You have to say, this is going to happen, right? Be very specific. Don't be like, oh, maybe this will, this is going to happen, and this is why we think this. Oops, sorry. Why we think this, or I think this. Right? So you're giving some sort of support piece to why you're saying what you're saying. It could be totally wrong. That's fine. It's not about being right or wrong. It's about being definitive, laying it out there, designing the experiment to hopefully answer this hypothesis, and then going for it. And you might be wrong, right? Your hypothesis might be wrong, but you still gain knowledge by being wrong, which is the important thing. So then you design your experiment, and we'll talk about this in the next slide. We want to have only one variable when we design experiments, at least at this point in your career. Because if you alter that one variable, then you can see how it responds. All right, so you might alter, we'll go through an example experiment here in a second where we might alter the fertilizer to see how a plant grows. And if you were altering the fertilizer and the water content to see how a plant grows, you might be like, oh, hey, this much fertilizer made the plant grow, but it was really the water that changed. So you only want to have really one variable as you go there. So we want to try to control the experiment as well. We'll talk about that. Now the data, there's two different types of data that you have. You can have quantitative, and you could have qualitative. God, it's hard to write with this pen. So quantitative would be your number components, and qualitative would be like a relative statements like, oh, it's hot, or it's cold, right? Or it's crunchy, or it's salty, right? Those types of things are qualitative. So quantitative, we could have, oh, it's 76 degrees Fahrenheit. Now, how are these different? Well, 76 degrees Fahrenheit is a number base, and qualitative, that's subjective, right? So it's good to gather both of these pieces of information, but oftentimes the quantitative one is the one that's going to go on a graph. So when we go graphing, obviously we have axes, so we put the y-axis here and the x-axis here. We're going to put the x-axis piece as the one that we alter, so the independent, I'll talk about this in a second, and the dependent we're going to put on the y-axis, and the dependent is going to respond to what you do in the experiment. From here on out, I would prefer you go after doing dots and then either connecting the dots or doing best fit lines, because this will give you the best understanding of what's happening, because this ends up showing trends, and that's the goal, is to find a trend or a pattern with your experiment. Okay, we'll do more graphing and more experimentation here in the very near future. So avoid the histograms, avoid the bars, right? I'm going to bar you from using bars because bar graphs are they might be easy to make, but they are not good for finding trends a lot of times. And they're usually ones where you don't have a chronological order components to your x-axis. And a lot of the stuff that we're doing in our class is going to be some sort of chronological component on the x-axis. And then we get to the creme de la creme, the conclusion. Conclusion is a wrap-up of what happened. And then you have to put your brain into it and explain why you think it happened the way it did. And we'll practice that more and more as we go through all of our future experiments as we go through the year. All right, so independent versus dependent. So independent, once again, x-axis, dependent, y-axis. The independent is what you changed oftentimes, right, or what's being altered. And then the dependent responds to your change. Right, so I kind of gave you an experiment that we were just talking about where we talk about plants. Right, so beautiful little plants. Woo. And we're going to test if fertilizer components will make this plant grow taller. That's a horrible plant. You can leaf if you want, all right? So this plant is going to respond to changes in fertilizer concentration. This is what you're changing. So the fertilizer concentration, concentration is going to go on the x-axis, right? So we might have a little bit, we might have medium amount, and we have a lot, right? Those are and we turn those into quantitative amounts like milligrams or something like that. And then the height of the plant is going to respond. Now what we might find is a little bit of fertilizer the plant might grow pretty good, right? And then as it goes a little bit more, maybe it gets taller with a little bit more, but then as you keep increasing it, all of a sudden, oh my gosh, it just dies off. So there might be a sweet spot in that component, but you wouldn't be able to assess that if you're doing bars a lot of times, because you wouldn't be able to see that trend because those pieces would overlap each other. So that's dependent and independent. Now, obviously you want to control as well. So with control, you're trying to keep every other thing the same. Things. Now, when I say other things the same, I mean like the amount of water, the amount of sun or light that you're using, the soil amount, where you plant the seed, right? All those things would be a control piece because we only want to vary one variable. And in this thought experiment that we put together, the fertilizer component would be the one variable that we're talking about. All right, on to the next. So you will have to definitely do blood type charts, right? So there's these two words that sound very similar, antibody and antigen. Antigen, if we look over here at these pictures, the antigen is the little flag that's sticking up here. So we've got on A blood, they have a little triangle flag, right? We kind of did it in class where, hey, this person has an A flag. That's the antigen. The flag is the antigen, right? So the B's, they have these circles, and then the ABs, they have triangles and circles, and then O's don't have any. So the O's have 0 antigen. The antibody is the thing that you make to fight the disease, right? So fight non-you, right? That's the easiest way I can explain it. So if you have A blood, you don't make A antibodies. You make the only other flag that's out there antibodies. You make B antibodies, right? If you're AB, you have an A flag and a B flag, so you're not going to make any antibodies. And if you're O, you have 0 antigens, so you make A antibodies and B antibodies, right? And then that prescribes into the RH as well, which RH would be another flag. You'd have another flag sticking up here possibly. If they're AB positive, it would have a little positive flag as well that goes up there. So let's look. On the test, you'll see something like this, which is things that we've seen on our quizzes in the past and also what we did back in lab. So individual 1, clumping on anti-A. This is antibody A. That means this person's antibody component, if it clumps, that means that they actually have A blood in that location. So since 1, it did not clump here, that means they don't have A. So this is not A, right? But on anti-B, it clumped, which means they at least have B blood in our knowledge base, right? And then we go to the last one, and it clumped on anti-RH. That means that they also have RH. So this person is B positive blood, right? If we go to individual 2, we see anti-A, it clumped on A. Anti-B, it clumped on B. And then RH, it did not. So this person has AB negative, right? If you need to hear that again, rewind, play it again. Hopefully that makes enough sense for you. So if you think about it, if a person has B blood, right, that means that they have a B flag. They're going to make A antibodies. So who can they take blood from? Well, this blood right here doesn't have a flag, so they could definitely take O blood, right? So they can take O blood. They can also take from B because it matches them. So this person, right, without looking at the RH factor, could take from B and O. Now, if you had B positive, then that makes things a little trickier because they can take blood from positives and they can take blood for negatives. But if they were negative, like AB negative, this person right here cannot even take blood from an A positive person because they're making antibodies against that positive flag. So that would be a no-go for a person that has a negative. Hopefully that makes sense. If it does not, then obviously email your teacher and hopefully can work it out with you, or you can come in and obviously ask questions while you're in class, okay? All right, so DNA structure, the next part of our story. So with DNA structure, you need to know, number 1, how it's put together, and then all the cool details that go with it. So the first piece is a nucleotide. So that is this phosphate bound to a sugar, bound to a nitrogen base. So you got to know that. So this sugar in DNA is deoxy, oh, that's a B, deoxyribose sugar. The nitrogen base is the part that varies. The phosphate always stays the same. These right here, those bonds, those lines are covalent bonds. They're nice and strong. So the nucleotides are not going to break apart, okay? The nitrogen base, that could be the A, that could be the T, that could be the G, that could be the C. Those pieces vary, and you can kind of see it over here in this model. Here's the phosphate, here's the sugar, and here's the nitrogen base. So T, thymine, is the nitrogen base. So this whole piece right here is a nucleotide. And then you look, there's another nucleotide that binds to it. So nucleotides then bind to each other through also a hard line, which means that those bonds that go down the side of the DNA are also covalent. All right, so there's one bond type is covalent. The other bond type that we see is down the middle of the DNA. So from one side to the other, you see these dotted lines. Those dotted lines are hydrogen bonds. Right? The chemist would say it's an interaction, not truly a bond. And that's fine. You just need to understand that this is pretty weak, which is awesome for our ability to open this up, right? You can pull the DNA apart and be able to get at the code, because the code of U is this TCAG blah, 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 all the way down code, right? So we have this incredible power of information that runs down the middle of your DNA. Now, when we address charge, charge is because this phosphate group has a negative charge to it, all of DNA has a negative charge, which is great because when we talk about electrophoresis, we'll talk about that right at the end, that piece, that the fact that DNA is negative, it will pull towards a positive if we set an electrical charge up, which is great. The last piece, the nitrogen bases, the A's, the T's, the G's, the C's, you need to know what's what. So the A and the G are the purines, and the C, U, and T are the pyrimidines. Why do we make you know this? Well, because a purine is always going to go with a pyrimidine, and they go in very specific connections. So G goes with C, and A goes with T. Awesome teacher, great class, right? So the G will go with the C in a hydrogen bond. The A will go with the T in a hydrogen bond. You can kind of see here, right? What's better, the awesome teacher or the great class? Oh, the great class, because there's 3 hydrogen bonds here versus 2. Awesome teacher, not to say that the teacher isn't awesome. Well, what about you, right? Well, in RNA, remember, it's not an A that goes here, it's a U. Or, I'm sorry, it's an A that's here. You're awesome, sorry. And you are awesome as well, not just the teacher, right? So that would be just in RNA, just so that we're on the same page. So then, taking that DNA, we can cut it with enzymes. And the enzymes that are going to cut are called restriction enzymes. So these are molecular scissors. They're DNA scissors that can cut DNA at precise locations. They are very specific. Every single restriction enzyme cuts in a totally different spot. So you can see here, EcoR1 is going to look for the GAATTC, and it's going to cut between a G and an A. So in this scenario, it makes a jagged cut, right? So you read it 5 to 3, 5 to 3, right? So you read it left to right, and then on the underside, remember, DNA is opposite, right? It goes opposite, so one side is upside down, right? You can see this here. If this is my T, top of the sugar, right? You can see it's going upside down here. Then we read it backwards on the bottom half of this piece. So EcoR1 cuts at that spot, whereas here, SMA1 is looking for CCC, GGG, and it's going to cut right between the CC, the last C, and the first G. So we end up getting this split piece. So far, we've only looked at blunt end cuts as opposed to sticky ends. We might talk about sticky ends in the distant future, but at least right now. So here's the cool part. Since everybody has different DNA, even though we're the same, 99.9% of our DNA is exactly the same, those little amounts that are different, that 0.1 that's different, causes these restriction enzymes to skip on some people because it's not the correct code for them to find. And so we call that restriction. Let me try to fit it in here. Fragments, because the pieces that come out are called fragments. Length, because everybody's lengths are different. Polymorphisms. So each of those pieces is slightly different size. Every single person's will end up having different riflips, right? They'll have different restriction fragment length polymorphisms. So here's your example, individual 1, individual 2. We're going to be looking for the cuts that we were using in class, which was the GAATTC, right? That's the one that we're going to look for. Was it the one? I'm trying to think now. Yes, I think that's the one that we're going to look for that we used. So let's look at where it would end up cutting. So between the G and the A, I'm going to cut right here. And we look again. So we have GAATTC. We keep looking. GAATT. Nope, not there. GAATTC. Okay, here's another one. So we're going to cut there. All right, so if you look, that's it. So we have one piece, two piece, three piece. So two cuts, three pieces. Okay, and then you look at an individual two. We keep looking. Man, this DNA looks almost exactly the same. Here's another cut. All right, keep looking. GAAT. Oh, that's slightly off right there. That A is not a T. We can't cut there. Oh, that's it. Okay, so that's 4, 5, right? So two individuals. We have only two pieces on this person. So this would be a rift flip. So this person has a different cut pattern compared to individual two. So then when we run this through a gel, next slide, it would end up showing up different. So remember, we load the DNA into the negative side. So if you remember, the gels had those little wells cut into them where you shot your DNA. So we're going to put individual one here. We're going to put individual two here. Now if we go back and look, individual one had 1, 2, 3 pieces. And I would say probably piece 3 is the smallest, piece 2 is the biggest, and piece 1 is the medium size. So piece 3, I'm going to put farther down here because it moved farther down the gel, because remember DNA moves from negative to positive because it has that negative charge because of the phosphate. So that would be piece 3. Let's look back again. Piece 1 would be a little bit higher up. And then piece 2, just a little bit higher than that. Okay, so that would be individual one's DNA fingerprint, right? So you have 3 bands from those 2 cuts that they have. Now you look at individual 2, they have this big, huge, massive band, and then they have 4, which is the same size as individual one's 1 piece. Oops, wrong direction. So they would have a piece right here, and we're going to call that piece 4. And then they have this big piece, 5, and it's bigger than all the others. And since bigger means slower moving, it would come out up here, closer to where we shot it in. So you can see that individual 1 and individual 2 have different DNA fingerprints. Well, why is this so important? Well, at this point in your career, what can we use gel electrophoresis for? CSI, crime scene investigation. So if we had DNA from the crime scene and we cut it with the same restriction enzymes, and we loaded it into the gel, and all of a sudden it came out, bloop, bloop. Oh my gosh, individual 2 is at the scene of the crime. Doesn't mean that they did it, but they definitely left some of their DNA at the scene of the crime. Beautiful. So that is what it's used for. We can use it for crime scenes. We can also use it for paternity tests, for looking at differences between different species, if we're going to do some evolution stuff. There's a whole host of different things. Finding diseases, if you know what the sequence is for the disease, like we'll look at sickle cell anemia in the future, and we'll do the similar type of thing for detecting a disease. But in this case, right now, here we have the crime scene, right? You can see 3 brands. This band up here would be the biggest. This one would be the smallest down here. And if you look, individual 1, individual 2, 3, 4, whose matches the crime scene? Bing, 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 individual 4. Now, even if it looked almost identical, but this band was missing, then they don't match. They have to match exactly the scene of the crime. And that is really important for you to understand, that it has to match the same. It can't just be, oh, well, a couple of the bands are the same, like individual 1. Oh, a couple of the bands are the same, so maybe one was kind of there. No, 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 no. It's either correct, it matches exactly, or it doesn't match. That's it. It's binary, yes or no, okay? So it's not a maybe situation. All right, so then one of the last pieces we did was we looked at body systems, and you're going to have to, on the test, give one structure and know the general function. Okay, so you're going to have to know, like, digestive system, stomach, break down food. Let me do this quick. All right, nervous system, you can have brain, internal communication, or responding, or anything that that matches up with the cards that you had. Muscles, you can have obviously muscles, and this would be to move. All right, skeletal system, you could have bones, and it could be for support and protection. Urinary, you can have bladder and kidneys. to filter out the bad stuff and balance your water in your body. Endocrine, you can have glands, pituitary gland, pancreas, all those different things, and that's to produce hormones to regulate things inside your body. Respiratory, lungs, gas exchange. Moving very quick because I noticed this is already 30 minutes long. Cardiovascular, heart. That's to pump blood, move blood around your body so it can get waste to its destination to get out of the body and move around the stuff that you need. Last piece, or last two pieces, HIPAA. You need to know that it's the Health, Insurance, Portability, Accountability Act, right? So the whole purpose here is to protect individuals, right? Information specifically about their health, right? Their health. So what is it? Who does it protect? The individual, right? And so so let's just say you had somebody, Johnny. I keep using Johnny. Johnny is 16 years old. Okay. Who has privy to Johnny's health insurance? Well, or health information, not insurance. His doctor would have information to it. Maybe the nurse that works with Johnny, but then also his parents, okay, because he is not 18. Once Johnny turns 18, his parents are off the list. Then it's just the doctors and the nurse. So violations, if a doctor is haphazardly talking to another individual that is not dealing with Johnny at all, and he's like joking about how, I don't know, Johnny doesn't do something, he doesn't brush his teeth or something like that, and they're joking and stuff, and they're sitting at a dinner somewhere, and the two doctors are friends, and they're saying this in public, that is a violation of Johnny's health information. And so these doctors, both that are involved in this situation, especially if this doctor is responding back to this situation, they both could be liable and be sued for that situation, right? So violations can be taken very seriously depending on how it is. And it's not just doctors and nurses. It's anybody that has information about another person's health, right? So this would include any like social workers. This could be teachers. This could be camp counselors, right? It's mostly to protect the individual, right? So if it's not helping the individual and it's violating that situation, that could be serious legal ramifications to that. Last slide. Manners of death. You need to know that there's 5 manners of death. Homicide, right, which is killing. Right, so you murder somebody, something like that. Accidental, that it wasn't intentional that happened. So you might say, well, was that drug overdose accidental or was it suicide? Well, if they were intending to get high and not commit suicide, then it's an accidental death, right? And that comes up a lot. So think about like drug overdoses. A lot of times are accidental. If a person has a heart attack, right? Well, was that heart attack because they did something else, or was it a natural cause, right? So we really got to take in a lot of information about that situation. If you don't know, then guess what it goes under? Oh, undetermined, right? So suicide, they're willingly taking their own life in that situation. So these are the 5 that you need to know. You need to be able to classify on a multiple choice. If I give you a scenario, what manner of death it would be labeled under. Now, with your Anna Garcia, you might have picked one of these already. Hopefully you already understood that it's probably not homicide. We need to narrow down some of the other pieces that go with it, and we will in all the other future units. All right, that concludes the review session. Hopefully that helps. If you have any questions, please talk to your teacher, email them, talk to them in person. I wish you the best of luck with your review. And I hope you destroy this test in a good way.